Sarah Wofford, and I'm with the biology department in the School of Science. I am going to introduce my graduate student, Alex Rocco, who's currently finishing up his thesis work. So what he's going to present today is a really cool project he's been working on for his thesis. And this is kind of the first round of data collection before he defends in March. So take it away, Alex.
Thank you for the introduction, Dr. Wofford. Like she said, my name is Alex Rocco. I'm a graduate student in the Behavioral and Sensory Ecology Lab here at JSU. And today I'm going to be talking to you guys a little bit about my thesis research, which is comparing aggression differences between a native and invasive species of crayfish. So the southeastern United States is widely regarded as a biodiversity hotspot. In particular, the state of Alabama is really well known for its high biodiversity. Alabama is known to hold 38% of North America's freshwater fishes, 43% of its freshwater gill-breathing snails, and 97 out of nearly 400 of North America's crayfish species. So Alabama crayfish have some of the highest species richness, or the highest species richness of any state or province in North America. And we have approximately 15 endemic species, which means they only occur in the state of Alabama. Crayfishes are also still being discovered, especially cave crayfishes in northern Alabama. We currently have 3 invasive crayfishes which have been identified in the state. And that's going to be Faxonius palmeri, Faxonius juvenilis, and Faxonius virilis. Invasive crayfishes have become a very big problem in recent years. Predominantly North American crayfishes are the most destructive invasives, and they're spread mainly through aquaculture as a food source, fishing bait, and through things like class pet releases or just regular pet releases. The University of Wisconsin recently did a study between 1979 and 2004 studying a Faxonius rusticus invasion at their Trout Lake Station in northern Wisconsin. And they found that snail populations were reduced from greater than 10,000 per square meter to less than 5 per square meter. Native crayfishes were nearly eliminated from the lake altogether. And the macrophyte richness was reduced by greater than 80% in some areas. And you can see this map of Trout Lake here. The invasion started in 1983, right here at this boat launch at Aliquosh Creek. And over the next 12 years, it spread entirely around the lake. And this is a problem that we see in all crayfish invasions, that their spread is so quick and so aggressive. And this is pretty true across all species, including the species I studied, the virile crayfish, also known as Faxonius virilis. They are native to the Midwestern United States and southern Canada, and they're invading waterways around the world. In Alabama, they're invading the Coosa River system, the Black Warrior River system, and the Cahaba River system. In the Pacific Northwest, they're invading the Snake River Basin, and then they're also invading waterways in Europe, particularly in the UK, the Netherlands, and Belgium. And one of the reasons why they're becoming such a dangerous invasive species is because you can buy these guys on Amazon. This is my Amazon page. This is Faxonius virilis. You can buy 100 crayfish for $90. So for less than $1 a crayfish, everybody here can ruin their watershed for decades to come. They're responsible for widespread habitat degradation, so they actually are known to use secondary burrows so they can damage flood control structures. They're also extremely ecologically damaging as overgrazers and predators. They're causing a lot of problems for a native crayfish around here called the reticulate crayfish, also known as Faxonius ericksonianus. They're widespread throughout the southeastern United States, especially in the Tennessee River Basin, the Coosa Basin, and the Cahaba Basin. And those of you who saw on the last slide, the Coosa Basin and the Cahaba Basin are both being invaded by Faxonius virilis. When we did our collection of Faxonius virilis in October of this year, we found 4 reticulate crayfish individuals, and we found over 300 Faxonius virilis individuals during that same time period. So they're being pushed back in a lot of areas in which Faxonius virilis has invaded. They're also extremely common in streams around here in Jacksonville, Alabama, so that made them a prime specimen for study in this invasive species study. So crayfish are widely used as a model for aggression for several reasons. They use chemical signals widely for communication when sizing up fights, and they can be used to signal dominance or submission during these fights. And those can also be manipulated in order to change the way that the fight plays out. They're also known to establish fairly linear dominance hierarchies, and when they are fighting, their fights are highly ritualized and they tend to be similar interspecifically. So you can fight multiple species against each other and expect similar behaviors between the two species. So for my crayfish treatments, I isolated them for a week beforehand to prevent any problems with social memory. We wanted these to be novel encounters. And each crayfish was marked with a whiteout pen with either an X or an O marking. And that ensured that the test was a little bit more blind, so instead of knowing which species was which, you only knew that this was the X crayfish or this was the O crayfish. So my first treatment was invasive versus invasive. My second treatment was native versus native. And my third treatment was invasive versus native. For my dyadic trials, I housed them in a 40 by 40 centimeter plexiglass arena with removable dividers. So you can see on this picture here, this divider has been removed, whereas this one is still intact. The removed divider allows for my crayfishes to interact uninterrupted, and the divider, when it's in place, isolates the crayfish from each other. It's blacked out to prevent them from distracting each other, and to prevent myself from distracting the crayfish. And when I actually ran my trials, I allowed the crayfishes to acclimate for 15 minutes. Just to get used to the arena, and then I allowed the crayfishes to interact uninterrupted for another 15 minutes. And I recorded the first agonistic interaction's duration and intensity. And then I used Bergman and Moore's ethogram shown over here to evaluate that intensity. And Bergman and Moore's ethogram assigns numerical values depending on the intensity of the interaction. Like I said, interactions are very stereotyped. So you can see we've got -1 and -2 over here, involving a tail flip away or a slow retreat. 0 being visually ignoring the opponent with no response or threat display. And then we have non-escalated agonistic interactions 1 through 3, in which they approach each other and can start boxing with closed chile. And then finally we have escalated interactions 4 and 5, in which they are actively trying to flip each other or tear or pull at appendages. So my hypothesis was that Faxonius virilis will demonstrate significantly more intense aggression than Faxonius ericksonianus in these dyadic fight trials. And I had a secondary hypothesis that these Faxonius virilis control trials would have significantly longer first bouts than Faxonius ericksonianus. So for my results, I ran an ANOVA on log transform duration data. The duration data was not a normal distribution, so I had to log transform it beforehand. And I used RStudio base package for those statistics. And I found that Faxonius ericksonianus had significantly longer bout durations than Faxonius virilis controls, with a p-score of 0.04. However, I did not find a significant difference between the interspecific treatment or either of the control treatments. But you can see over here on this box plot that the ericksonianus control treatment had longer trials, and then there's a sort of stepwise pattern down to the virilis, which I found extremely interesting. My Tukey post hoc also confirmed the significance of the control bout duration differences with a p-score of 0.0401. I also ran a chi-squared test to determine the significance of the escalation of the bouts, and I found that there was no significant difference between escalation of bouts. However, you can see over here on this bar chart that the ericksonianus control had 7 non-escalated bouts and 2 escalated bouts, whereas the interspecific had 7 escalated bouts and 2 non-escalated bouts. So I believe that larger sample size may actually change the significance of this data, and that's just something we'll have to wait and see for as I run more trials. So going back to my hypothesis, I had to reject my first hypothesis that Faxonius virilis would demonstrate more intense aggression during these fight trials. In fact, there was no significant difference between aggression and fight trials. And then I also rejected my secondary hypothesis, which said that Faxonius virilis had significantly longer first bouts. In fact, Faxonius virilis had significantly shorter first bouts than Faxonius ericksonianus. I also had some observational data which I couldn't use any statistics for, but I felt was important to note. I had 2 non-fatal mutilations which occurred in my interspecific trials. Both occurred to Faxonius virilis. One lost a chela and a rear leg, and one lost a non-moving finger and an antenna. Of my 10 interspecific trials, it was generally a wash. There was no clear winner between them. And then I also had 2 Faxonius virilis trials that were thrown out due to no interactions, and 1 Faxonius ericksonianus trial that was thrown out due to the fact that there was actually no interaction conclusion. So they actually fought for so long that I had to conclude my trial before it finished. So what are the implications of my study? It seems that Faxonius virilis can form dominance hierarchies faster than Faxonius ericksonianus, which actually may lead to them being less susceptible to predation. So if they're taking less time to be out in the open and fight each other, they may actually be less vulnerable to predation. And my interspecific bouts, again, may be more significant, may be significantly more intense with further trials, so we'll have to wait and see on that. And then Faxonius virilis does not show higher aggression than Faxonius ericksonianus, which means we need to look in different directions if we're going to determine the cause of this invasion, be that either fecundity or how they respond to predators or any other variable we may have to look at. Finally, I'd like to acknowledge the Jacksonville State University Department of Biology, specifically the Behavioral and Sensory Ecology Lab, the Kinsolv Travel Grant, and Bowling Green State University's Laboratory for Sensory Ecology. I also have photo credits available on request. Does anybody have any questions? DJ?
Yeah. So when you were running your trials, did you? And you may have said this before. Were those single-sex trials? Like, were they all males?
Yes, they were all Form 1 males. It's important to make sure that they are single-sex because some of Dr. Wofford's papers have shown that intersex matches can be different than single-sex matches. Any other questions?
Well, you said more, how many bouts did you run on these?
As of this presentation, I had an N equals 9 for my controls and an N equals 10 for my interspecific trials. But I'm currently running more trials right now. So I think I have N equals 15 about for each of my trials at the moment.
Where did you get each set of crayfish from? Were these from local rivers? Or?
Yeah, so the Faxonius virilis was collected in the Cahaba River, specifically Masonic Park in Trussville, Alabama. And then the Faxonius ericksonianus was actually collected at the Haynes Branch behind Cooter Browns and by Chief Ladiga Trail here in Jacksonville. Anything else? Dr. Ager?
So your original hypothesis, you had thought, if I remember right, was the invasive you thought would have a longer duration fight. Was there a reason you thought that, or just?
So there are there's a lot of data out there that suggests, generally speaking, that one of the reasons why invasive crayfishes are so prevalent and can dominate watersheds very quickly is because they're a lot more aggressive. So I expected that the bout durations would be longer because of that higher aggression seen in primary literature. Another one? Yeah, what's up?
Is there a size difference between two species? Or
These were size matched. One thing that's interesting to note is that it seems that Chile size, so their claws get larger in Faxonius ericksonianus quicker than Faxonius virilis, but I was size matching them within 10% of each other. Any other question?
I think I figured